Okay. Thank you, Antonio. You can begin.
Good morning, everyone, and I wish to thank MGI for inviting me here today and all of you for joining this webinar. So, as Brett already said, today I will show you how we are trying to formulate innovative immunogenic treatments in pancreatic cancer that we hope in the future they can change the landscape of pancreatic cancer treatments. So in this presentation, we will explore the findings from our recently published studies, in which we use Stereo-seq only to map the immunogenic effect of some of these novel therapeutic strategies. So this is the layout of the webinar. So first, I will give you just a brief introduction of pancreatic cancer and murine cancer models. Then I will show the first study focused on radiofrequency ablation, and the second study that is focused on immunocytokines. And then after, in the last two slides, I will just give you a brief overview of all the bioinformatic analysis that can be possible with the Stereo-seq only. So why it is important to study pancreatic cancer? So pancreatic cancer is a really lethal type of cancer, and it is an increasingly prevalent disease. So recent studies show that it's no longer just a geriatric cancer, but as young individuals, especially women under 35 years old, are being diagnosed at higher rates. So we face several challenges when we have to treat pancreatic cancer. The main one probably is the late diagnosis, because usually pancreatic cancer patients are diagnosed only at advanced stages, because usually this type of cancer just gives some vague symptoms like abdominal pain or back pain. And moreover, pancreatic cancer cells are really resistant to chemotherapeutic drugs. So even though in the recent year we did a lot of advances on surgery of pancreatic cancer, and also neoadjuvant therapy really create a breakthrough, and we managed to increase the rate of overall survival of patients with an overall survival measure of 5 years. However, most of the patients will die in less than 1 year. And as you can see here, this is the timeline of all the treatment that are now used in the clinic, and the option for the patient are really low, and are really not new. So there is an urgent need of novel therapies. One of the major hurdles in treating pancreatic cancer it's its immune deserted nature. So in this picture, you can see a typical topography of pancreatic cancer, in which you will have few pancreatic cancer cells that are surrounded by this dense stroma that is called the desmoplastic stroma, that protects the cells from the chemotherapeutic agents, but also from the immune cells that cannot infiltrate and kill the tumor. Also, in a recent study, we show, if you want to know more about this, you can see our recent study, in which we show that the aberrant hydroxyprolylation of this collagen create this tight stroma that exclude the immune cells from the tumor core. Moreover, where immune cells are present inside the tumor core, most of these immune cells are actually M2 macrophages and myelodysplastic suppressor cells, blood-derived suppressor cells that that exert the immune suppressive function and exclude the cytotoxic T cells. So the main aim of our research is to turn this desert into an oasis, so that we can lead the CD8 T cells, the cytotoxic T cell, but also natural NK cells inside the tumor to exert their natural anti-tumoral function. So today I will show you two different approaches that we treatment approaches that we use to evoke this change inside the tumor microenvironment, and and using these immunogenic approaches, we want to create an immunosuppressive environment to enhance the treatment effectiveness. So before proceeding with the studies, I just want to give you a brief introduction on the preclinical model that we use in these experiments for these experiments, because so you can understand better all the different types of experiments that we did. And the most used type of mouse model that is used in pancreatic cancer studies is the KPC model, that is a genetically engineered mouse that harbor KRAS and TP53 mutation. They are the founding mutation of pancreatic cancer. And this model works really well, and it can recreate all the different histopathology of pancreatic cancer, and also all its oncogenesis from from low panine to high grade pancreatic cancer and metastasis. So what we did is that we established the cell line from different KPC mice, and we then characterized them for the ability to recreate the different clinical and molecular features of pancreatic cancer by RNA sequencing and immunofiltrate analysis. So what we found that all of these models can recreate all the different subtypes of pancreatic cancer that are usually are found in the human counterpart. So in the human counterpart, the pancreatic cancer is usually are classified in different with different classification, different name. Most of them can be divided in this binary classification of classical and basal, where the classical is the low-grade type of cancer that is more differentiated and also had a better prognosis, while the basal-like and the squamous-like represent the high-grade type of cancer, and they're also associated with a poor prognosis. And so as you can see here, we established this six model that reflect not only of the different subtypes of human pancreatic cancer, but also the different clinical feature and their uh, survival feature. So we also did something that is very important for our study. It's an immune characterization of these models, and we did an immune filtration profiling, and we identified two models that represent the typical the immune desert in pancreatic cancer, in which there is really absent of really low immune filtration. Then we established two models with low immunogenicity and two models that were really immunogenic. So if you want to know more about this, you can check our article here. So leveraging these models, we can study the effects of different immunogenic treatments in a setting which we know all its molecular and immunological features. So this is the first study of the day that we did in collaboration with Francesco De Santis from University of Verona, and as I told you, in this study, we focused on radiofrequency ablation. So radiofrequency ablation, or RFA, generate a localized heat that destroy the cancer tissue. Usually in pancreatic cancer, this radiofrequency ablation is done by ultrasound guided endoscopy, and this electrode is inserted in a small electrode that is located inside the tumor, and produce using an ion stream a really high heat measure of 60 degrees. So and this high heat kill tumor cells. However, until now, RFA there is a limited use of RFA in pancreatic cancer patients, and usually only a few patients with a resectable local advance uh, disease will undergo under RFA, also with scarce results, and most of the cases used for palliation of symptoms. But we want to study its immunogenic effects and how we can use it to formulate a novel treatment strategy to enhance what are now the limited therapeutic options in pancreatic cancer. So we started with a simple experiment in which we exposed our KPC models to hyperthermia, so to 56 degrees for 30 minutes. And of course, we expected the cells start to die. But most importantly, we found that they release HNGB1. This protein acts as damage-associated molecular patterns and is a pro-inflammation signaling molecule that activate the immune response by T receptor like the Toll-like receptor and RAGE. So when these cells are treated, they release HMGB1, and then we co-culture this treated KPC cell with immature dendritic cells, and we found that this hyperthermia evoke a really increase in the activation of these antigen presenting cells. So after we did an in vivo characterization of the RFA immunogenic effects, so what we did is that we did an orthotopic model with the FC1242, that is one of the KPC model that I showed you before, that is characterized by a classical subtype and also a low immunogenic potential. And we found that the FAA induced a consistent necrosis in the tumor core, and then we performed a tumor immune profiling by both cytometry and the Stereo-seq, and we found that after 48 hours, the RFA can evoke a really strong inflammation inside the tumor with by both the RNA-seq and cytometry, and by cytometry we found an infiltration of neutrophils that was really high, while we found no consistent upregulation of CD8 cells. After 10 days, we found that this inflammation is partially resolved, and we found only an infiltration of dendritic cells inside the tumor coats, while we found no presence of CD8. So on the base of this study, we also did a systemic immune profiling, and we found that almost the same results that we found in the tumor core can be found also systemically, where we found an increase on neutrophils and also an increase on neutrophil to lymphocyte ratio. And this is the typical sign of activation of an inflammatory signal. And this was also corroborated by the expression in less than 6 hours of high expression of HNGB1, but also other molecules that are involved in inflammation. Moreover, we also found a consistent activation of APC, so the antigen presenting cells inside the spleen of these mice. So on the basis of this study, we can understood that this RFA can induce consistent damage on cancer cells that trigger the inflammatory response, and also the activation of all the antigen presenting cells. But we did not find any presence of CD8 T cells inside the tumor. So we hypothesized that this RFA can really synergize together with immunotherapy. And to do that, we did this time an heterotopic model So we injected the same model that I showed you before inside the flank of the mice, and we treat this tumor with immunotherapy, RFA, and the combination of both. And as you can see here, only the combination of radiofrequency ablation and immunotherapy could really reduce the tumor volume and also increase the survival. To confirm that RFA induced the activation of a tumor-specific T-cell response, we rechallenged the mice after 2 months from complete tumor elimination, and with the same KPC model, FC1242 in one flank, and another cell line in the other one. And as you can see here, we found a significant difference in only in the tumor volume of the FC1242, only in the mice treated with a combination of A and immunotherapy, while on the other side, the other cell line completely grow unchecked. So this means that the type of response that we can evoke using both RFA and immunotherapy is quite specific of the tumor and can trigger a long immune memory. So we then characterize again the effect of the combination of both RFA in both with monotherapy and in an orthotopic model. And as you can see, the results here is a little bit different because the orthotopic model is subjected to a much worse prognosis. But as you can see here, we have almost a complete reduction of the tumor if compared to the control. So now we arrive to the core of this webinar. And what we did is that thanks to the early access program, we decided to analyze two tissue from one control mice and one mice that was treated with RFA and monotherapy. And as you can see here, thanks to this technology, we were able to characterize all the different immunological and cancer cell regions that characterize the two different types of tumor. And we found really interesting results thanks to this technology. So we found a total novel pancreatic cancer cluster in our sample, and what was really striking for us that in the control were present more the cluster cancer cell clusters present in the control were more associated with the squamous or basal profile, so the types of cancer that are most aggressive, while in the tumor that was treated with a combination of RFA immunotherapy, we found that the squamous type was completely eradicated, and we found that most of the tumors present there was actually associated with a classical subtype. So this was really important for us because it showed for the first time that this RFA and immunotherapy combination can reshape not only the tumor microenvironment but also the cancer cells itself. And then, of course, we did the spatial immune profiling, and we scored the gene set activity for the signature associated with the anti-macrophages, inflammatory macrophages, and also with the different types of APC and activated T cells. And what we found is that while in the control, uh, the cells, the immune cell present were mostly M2 macrophages and inflammatory macrophages. In the treated tumor, we found that almost all of the immune cells present were APC and activated T cells. And what was really great that we could do with this technology is that we mapped the exact region in which the tumor specific response is generated. So we found this region that was near the tumor, that was rich in antigen presenting cells and thymocyte T cells. And TEMIS has an essential role in CD8 T cell activation and modulates the T cell receptor signal activity, ensuring an effective antigen recognition and optimize the cytotoxic T cell response. So thanks to stereosectomy, we can able to find the exact spot in which we evoke our immunogenic response. So after these nice results, we also confirmed with the spatial results with immunofluorescence and immunohistochemistry. And so as you can see in the control, the CDA are completely absent and they increased with the combination. And we found a consistent activation of, uh, um, of immunosuppressive cells in the control that are not present inside the combo. So in conclusion, we showed that RFA cause a rapid and local inflammation of pancreatic cancer, and that the RFA immunotherapy can reshape pancreatic cancer cells into a less aggressive classical-like type. And we also showed that the combination of RFA immunotherapy create a tumor-specific T-cell mediated response that is mediated by both APCs and TMSI um, expressing T cells. So these findings we hope that may may pave the way for novel treatment strategy for resectable pancreatic cancer patients. So now we move to the second study of the day, and on this study that is from our group, and we did this study in collaboration with Philogen, and we focus on immunocytokines. Immunocytokines are a class of engineered fusion proteins that combine the specificity of the antibody with cytokines. So we, in this study in particular, we focus on ADBFN1 immune cytokines. The cytokines that use the L19 antibody that is able to target a specific isoform of the fibronectin that is present almost only in the cancer front, and especially in the tumor core, where it's not present in the other tissues. So for this study, we wanted to characterize the effect of these L19 cytokines harboring different types of cytokines like interleukin 2, interleukin 12, and TNF alpha. So the first thing that we did was that we evaluated the expression of this isoform in both organoids by both the immunohistochemistry and qPCR, and we confirmed the targeting ability of L19 antibody in our organoids that were originated from our qPCR model. So after this step, what we did is that we established a co-culture platform consisting of organoids and for this culture we use for this co-culture we use two different qPCR model, one with a really absent immunogenicity and one with a low immunogenicity. So then we treated these we co-cultured these organoids with T cells. What we found is that when we use the 19 interleukin 2 antibody, we found a significant difference in the activation of T cells, infiltration, and T cell and and cancer cells death. That is the green one that you can see here. That is reported that. Associated with the caspase activation and therefore apoptosis. After we check for again for the expression of this IDBFN1 and L19 targeting inside the tumor models. So these are all orthotopic models for different KPC cell lines. And as you can see here, this fibronectin isoform is present in almost all of our pancreatic cancer model, and why it's not present in all the other tissue. So the L19 can really um, lead interleukin to all the cytokines inside the tumor, avoiding side effects in the other tissues. So for our study, we used the KPC-06 organotypic syngeneic mouse model. So this model was characterized by a low immunogenic potential, and we treated these orthotopic models with the standard therapy and different types of L19 immunocytokines. And as you can see here, we found a complete different effect on tumor volume of the immune cytokines if compared to the standard therapy and also the control. And in particular, we were able to achieve complete cure with a high dose of L19 interleukin 2. So, using on the basis of these promising results, we formulated a treatment strategy that may be also used in a clinical practice. So, as I told you before, most of the patients are diagnosed in the late stages of the disease where the fitness is really low. So we want to use a low dose L19 interleukin 2 in combination with the FOLFOX, that is usually the standard therapy in the second line. So as you can see here, also at low doses, the immunocytokines could evoke a significant reduction in tumor volume and in combination with FOLFOX and a consistent um, increase in survival in both in KPC-06, that is a model that is characterized by a classical subtype, so not really aggressive but a really low immunogenic type of potential. But it works really well, especially in KPC-12, that is a KPC model that is characterized by a basal subtype, so it is the most aggressive type of cancer, and also is characterized by a really absent immunogenicity. So here again, we want to understand what was going on inside the tumor core when we use these different treatments. Here again, we use the Stereo-seq, and as you can see here, the results speaks for themselves. So we were able here again to find all the different clusters and regions that characterize the different types of tumors. And as you can see here, we found the filtration of CD25, CD122 cells inside the tumor only when we use L19 interleukin 2. So these are the immune cells that are activated and that express the L2 receptors. And most importantly, we found that there is a consistent increase in the presence and infiltration of NK cells and CD8 T cells and APC inside the tumors when they are treated with the L19 immunocytokines, but also with L19 immunocytokines and FOLFOX. So, in conclusion, in this study, we show that this ADB isoform of fibronectin is specifically expressed in pancreatic cancer. We also show that L19-based immunocytokines at high levels can cure KPC models, and we also formulated a low dose L19 interleukin 2 standard therapy that can be used in the clinics. So our institution will soon initiate a trial involving patients with different types of gastrointestinal cancer to evaluate the curative effects of both the high-dose L19 interleukin 2. So now we finish with the study. So before finishing with the webinar, I just want to give you a brief overview on all the bioinformatic analysis that are possible with the Stereo-seq. So, of course, when we are using Stereo-seq data, StereoPie, I think, is the first tool that can use, and this is tool that was specifically built on Stereo-seq data, and here you basically you can do almost all everything that you may need in for your analysis from Gaussian filtering to clustering to differential expression analysis, nebula analysis, and many other. But if you are more you like more R and Seurat, like me, you can also convert thanks to StereoSpy, you can convert the standard Stereo-seq data to a separate object, and thanks to this conversion, you have access to a huge number of spatial transcriptomic packages and especially you have access to the most advanced algorithms for data integration by traffic correction, the signature scoring. These are the one that I use for our studies that I show you today. And then of course you can use different all the different packages that do the cell annotation and spatial enrichment and niche and cell-to-cell communication analysis. So with this, I finished the webinar. I just want to thank all our collaborators and my group for all their help and for the data that are present to you today. And I also wish to thank all the funding and partners for these studies. So thank you very much.
So thank you, Antonio, for that insightful talk and for your presentation and just guiding everyone through your research as well as what to expect coming from your institution. And then also just showing that you can also, there's also the ease of the bioinformatics utilizing Stereo-seq Omni. So I would like to ask, there's a question, Agostini. Just give me one sec. So in the study, you used mouse as the sample type. Have you tried using human samples? Or are you also looking to use human samples, especially when you're looking into the clinical side? And then this question then can also be go to Javier about what are the species compatibility with Stereo-seq Omni?
So yes, we use also the human samples and it works really well. And we also managed to do microbiome analysis on these human samples. So yeah, you can do it really well. So it
Okay.
works really well for us.
And the bioinformatics using the human samples, is it the same? Is it the same
Yeah,
workflow?
see the same as I show. So basically, what I show you have one workflow that is focused more on Python and Python language and all the ScanPy modules. So the people that analyze these types of data know what I'm talking about. But I use a lot also. I prefer to use Seurat and R, all the different biocontractor packages that I'm more familiar with. So also thanks to Javier and all their colleagues in MGI, I could convert all these Stereo-seq data, so the standard Stereo-seq data to a separate object and do all the analysis that I show you here. So you have all the options to do it.
Yeah, thank you, Antonio, for the very nice presentation. I think this really good, comprehensive, talk and guidance through the papers. Regarding to the question, Brett, we with the stereo-seq only can analyze any type of sample, so it is a species agnostic. So the way that we have to capture the RNA molecules is random, so is untargeted, and then this is very easy to move from one species to another. It might require a bit of optimization, but very limited because even the permeabilization time is fixed, so it's totally doable to do multiple species study. Yeah.
And with the FFPE?
This is a Stereo-seq only is actually designed for FFP samples because the FFP sample, the fixed samples, usually will have like some degradation of the RNA, and then you have like a fragmentation of the RNA molecules, and then the traditional way we use that is a poly A poly T base to capture the transcript is very limited in the efficiency because you will have many fragments that will have no the polyethylene. So we designed this new capturing method with a random probe that will capture all the RNA molecules depending whether they have the poly or not. So it is compatible with the FFP samples.
We have another question here. Do you find the high resolution of Stereo-seq useful? Do you actually use the smaller bins? And then maybe, Antonio, if you can also just mention what was the resolution that you were able to achieve or that you've seen with Stereo-seq in your study?
So the study I use B100, B100. So, but you can do it lower resolution, yes. But this was also not even the version one. This is the early access version. So I think I know that now works much better, right?
Yes, that's correct. So just to put in context, the earlier access for the FFPE that Antonio was participating didn't have the possibility of doing an image of the tissue section that now is possible with the only product. So you actually can reach to the single cell. So you can have cell segmentation based on the image, and then you can create a cell mass, and then you can go and look for the the genes that you have per cell, so reaching the single cell level. In the case of the data we get for this research, because it was earlier access, I think the we could sequence more. So the saturation for the sequencing was still like very low, and then at the end we decided not to sequence more because we found what we needed, right? Just increasing the bin size, as Antonio says. So they're using the bin size that is bin 100, that is like 50 micron. So, but this is not the best resolution you can get in their case because they have not enough genes per bin. If they reduce the bin size, then they decide to use a bin 100. But honestly, we will resequence that data and we will get more genes per being, right? We can reduce the resolution even without the cell beings and reach something more reasonable, like being 50, for example. That could be definitely achievable with this library that we generate, right? And the thing is that if you don't have the image, then you cannot reach it to the cell beam. So for this particular data set, was not possible to reach to the single cell, right? Even if you sequence more. Now the new version or the final version of the Stereo-seq only, I have been seeing data, and you actually can look in the Stereo-seq website where you have some like data set that you can see, including mouse brain, and then you really can reach to the single cell, and you can have around 400 or few hundreds of genes per cell. So this is the type of resolution that we can achieve with a Stereo-seq only.
And then with the chip sizes, does it matter? So what are the chip sizes that are available, and does the chip sizes also affect the resolution you can achieve?
Well, I think the chip size won't affect the resolution, but it could be convenient for different application. We actually are thinking, Antonio, to do some testing with a bigger chip size. As today, we only have one chip cipher only. There is one by one, but we're gonna release very soon different size similar for the chip T. So we're gonna have like zero point 0.5 by 0.5, 1 centimeter by 2 centimeter, 2 centimeter by 2 centimeter, and 2 centimeter by 3 centimeters. And we plan to launch it in the second half of the year to have this available. And then the resolution will be independent of that. We actually starting the production of our chip with a big one, like this 13 by 13. And basically what we do, we cut them down in small pieces. And the thing is that you need to have some QC and way to cut in the big chips to be able to launch the bigger size chips, right? But on the customized basis, you actually can make bigger than 2 by 3, right? So that would require like some longer time to produce the chips, and but it will be possible to do even bigger than 2 by 3 centimeter.
So, Antonio, we in one of the polls we've asked what feature would be the most important to someone when considering spatial transcriptomics, and they mentioned the ease of workflow and analysis. Would you say for your lab to adopt Stereo-seq or spatial transcriptomics, is this one of the, let's say, the most challenges that you see in your lab to adopt this type of technology? Or do you think it's the resolution and sensitivity, the sample type flexibility, or cost effectiveness? Or do you think the workflow and analytics is probably the most important?
Well, that's that's a good question. So actually, we use several types of spatial techniques. So that much depends on your lab, on the expertise of your lab, and also all the different equipment that you have. So I haven't done much the workflow because our data was generated in Riga lab for
Mhm.
Stereo-seq, but we do all the other stuff. So usually, what we really like of Stereo-seq was the possibility. Of course, it was to segment everything. Of course, segmenting everything, capturing everything, it can be a hurdle, and we had at first it was challenging, but on the other, after you have a lot, you also have a lot of information on which you can work. So I can speak about the informatics side. So Stereo-seq was it's quite so you can start with StereoPy. So it's a really good suite. I mean, you can do all the analysis. It's quite easy, but we had, of course, especially in the first study that I showed you, we had a specific question that and a specific no event that we have to map. We had to show and prove that there is these APC that were near the cancer and they activate the cell and engage them against these cancer cells. So of course to do that you need more analysis, and of course you can rely on you can integrate Serrat together. So actually we integrated the StereoPy with Serrat and also other different types of packages.
Hmm.
But of course,
So,
like the spatial analysis is not an easy task. So yeah, it
Yeah,
takes.
if I can follow up on the question. So, do you think the fact that now you can also see the microbiome and the no coding RNA like useful for your
Yeah,
research?
yeah, of course. As I say, you're segmenting. You can segment many things. So of course you can just data on whatever you want. I mean, of course, on one side that makes the clustering a little bit much difficult, but it's a different methodology, a different kit, a different type of data, and you need times to work on it. But in the end, we made it. So we already published, thanks to IBR, this data. So
Thank you.
So yeah, we see that many people were saying that a lack of technical expertise is one of the biggest challenges that they see with spatial. But I'd like to mention, as what Dr. Agostini's also said, is Javier is the person that can help support. So Javier, if you don't mind, after the webinar, I will share with everyone the on-demand link, and then also just share your information if you don't mind. And then they can reach out to Javier. So,
Absolutely, but I want to highlight here that the capacity for Antonio to analyze the data was really very fast. Sometimes this is critical point that you have data, and it takes a long time for analysis. I think on Antonio's hand, actually, it was actually really fast to get from the fast queue to the process data. This is very important to have the as we say the expertise, right? So they do have the expertise in spatial and atomic data, not from Stereo-seq, but they have it, so they really could master and getting the best of the Stereo-seq data really quickly.
Antonio, you mentioned that your lab or institute is using various different spatial
Yeah,
technologies.
yeah.
Are you combining each technology to then come up with a better answer to the questions that you're asking, or are you more focused now to use one technology over the other? How are you managing this? Yeah.
No, I just managed. I mean, I use the technology. So the most important thing with spatial, I think, is the question that is behind, because
Yeah.
why you need to use spatial. So if you just want to do spatial with some samples, maybe you will not have great data. So the question is the most important part. And also, yes, it depends what type the question you have. You can use. So, for example, here we have the Xenium that we can use. You can have a small panel but analyze a lot of samples at single cell resolution. Or, of course, you can use Stereo-seq when you want to check things that are not present in, for example, the Visium that is pro-based, but you can sequence everything. You want to see the microbiome or everything else without bias. So it depends much by the question. So we are planning on using again Stereo-seq, especially in the, as Javier said, on the bigger chip, and we will see later. So maybe we will have another webinar in some years.
So,
Perfect. Okay, thank you, Antonio. So with Stereo-seq, obviously, you can answer a lot more questions and interrogate tissue samples or complex tissue samples a lot more. So I don't know if there's anything else, Javier, from your side you want to mention?
no, I think everything is very well covered. I just want to add a comment that for discovery, when you're gonna really want to do a discovery, and then, or maybe you already have an RNA molecule that you might be interested, and this is not including in the panels from the like targeted method, then a COC could be very useful because you can get a quick answer or a quick exploring of your question in your sample without needed to give the information to the third party of the sequence of your RNA molecule, right? And that can speed and facilitate a lot of some of the process that otherwise would require NDA and a lot of work for generating the new props. I don't know, Antonio, if you could comment on that. What are your thoughts for these type of situations?
Yeah, I mean, yeah, that's I mean the major advantage of this technology, as I say, is this: you can sequence everything, understand everything, and do your best to analyze it, and uh, uh, and find whatever you may need for your project. So yeah, that's the that's probably the most important things that I say always. For doing spatial, you need to have a really special question, real question, and then choose the best option that you have. So,
Cool. Well, thank you, Antonio and Javier, for being with us today. And thank you, everyone, for joining online. We will close the session. We will follow up with another email for when we publish the webinar on demand, so you can view it in your free time. And if there's any questions, you can just reach out to us. Antonio, if there's any questions, we'll just get your information and then they can send questions as well.
yeah, of course.
Okay. But yeah, thank you so much, Dr. Agostini, for your
Right.
wonderful presentation. And we will see you again once
Yeah,
you've done more, more with the new chips. So great.
yeah.
Cool.
Thank you.
Thank
Well,
you.
thank you.